Hello. Today we're talking about 6.3 and 6.4, the nucleus and the endomembrane system. This is sort of the meat and potatoes of the central dogma. So remember that DNA, RNA, protein. So we're going to start talking about where the DNA is in the nucleus and how it gets transcribed and translated into a protein. And so the nucleus and the endomembrane system are very key components in this process. And so if we start looking at the nucleus. The nucleus, as we know, contains the genetic material, our DNA, if we're talking about a eukaryotic organism or even a prokaryotic organism, we're going to be talking about DNA. What's interesting about the nucleus in a eukaryotic organism is that it has a double membrane, so it has two lipid bilayers. This is true for a number of the other organelles. So there is an inner membrane, we call that the nuclear lamina, and it's composed of a bunch of proteins and intermediate filaments. Intermediate filaments are a part of the cytoskeleton that are made of keratin and exist longer. So this is a real sturdy part of the cytoskeleton that gives the cell its structure, and it exists in and around and near this nuclear lamina. And we can also see that there are pores in the nuclear membrane. This is going to be surrounded by protein structures that that pick and choose what can go into and out of the nucleus, because only very few different types of ions and molecules can get into the nucleus, because we need to protect that DNA. We don't want to alter gene expression or to change the way that our proteins are going to be created. And so the genetic material is separated into discrete units called chromosomes. So we humans have 46 chromosomes, 23 from mom and 23 from dad. And they are wrapped around these little structures here that we call histones. And so the more tightly that the DNA is wrapped around the histones is going to limit the amount of expression that can happen, or the ability for proteins to read the DNA. And so if it's wrapped tightly around these histones, it's called a chromosome. And as it relaxes and it is less tightly coiled around these histones, it becomes chromatin. And so during different parts of the cell cycle, the DNA is in a chromatin state, where it can be accessed by proteins or enzymes to read it and to transcribe. And so that is the difference between chromosomes and chromatin. Inside the nucleus, we have the nucleolus, which is where ribosomes are composed. And ribosomes are a really cool, interesting structure, not technically an organelle. They're not a membrane-bound organelle. And so they are composed of rRNA and a protein complex. And so they are made inside the nucleolus, which is inside the nucleus. So about 1/3 of the nucleus is the nucleolus, which is where ribosomes are composed. So after they are composed, after they are made, then the ribosomes are going to leave the nucleus, and they're going to either hang out in the cytosol, or they're going to hang out on the rough ER, the rough endoplasmic reticulum. And there they make proteins. They're going to read the mRNA and make proteins, or polypeptide chains. So the genetic material is in the nucleus. Remember the central dogma of biology. This is going to come again and again and again. We have DNA, RNA, and protein. So that DNA is where the information is stored. It gets transcribed into RNA by a protein or by enzymes, and then it leaves the nucleus and it is made into a protein with the help of a ribosome. So function follows structure, another important concept to remember. If a cell needs to produce many proteins or many enzymes, an example of this would be your pancreas. Your pancreas makes lots and lots of enzymes and lots of proteins. Well, then in those cells, you're going to have a lot more ribosomes present because it needs to make more proteins, so you're going to have more of the structures that make proteins. So we have left the nucleus and we are looking at the endomembrane system. This is going to consist of our nuclear envelope which is attached to our endoplasmic reticulum. We have smooth ER, we have rough ER. We're going to look at the Golgi apparatus and lysosomes and vesicles and vacuoles. So all of the little bubbles that help transport materials is going to be included in our endomembrane system. And so the function of the endomembrane system is pretty diverse, but we're going to synthesize and transport proteins. It works for metabolism and the detox of poisons. So if you look here, we have many small compartments, which means that many different chemical reactions can be occurring all at the same time. And we're going to look at the endoplasmic reticulum. So here, it's a network of lipid membranes, and they are connecting sort of tubules. And when you are on the inside of these tubules, you are in the lumen of the endomembrane system. And when you're on the outside, you're in the cytosol or the cytoplasm. So when you're in the inside, lumen often means the inside of something. So let's look at the smooth ER first. This is where we don't have ribosomes. And the function of the smooth ER is to produce lipids. It is the metabolism of carbs, so it breaks down carbs, and it detoxes the poisons that we put into our body, and also stores calcium. So the detoxification, usually what happens is your smooth ER is going to add a hydroxyl group, an OH, onto a molecule. So it makes it more soluble, and then your body can work to get rid of it more quickly. So if you have more smooth ER or a more effective smooth ER, you're going to have an increased tolerance to drugs and to alcohol. And so we also have the storage of calcium ions in the lumen, and this is used for muscle response and also for sending messages through your sensory cells, which we will talk about in depth pretty soon. The rough ER, this is where our ribosomes exist. This is what makes it the rough ER. And it is attached, you can see these little dots here. So when a mRNA goes to a ribosome that is attached to the rough ER, what's going to happen is that the protein is going to be made inside the lumen of the rough ER. And so when that protein leaves the rough ER, it's going to have that bubble of a lipid bilayer around it, which will allow it, kind of gives it that key to leave the cell. So the proteins that are going to be leaving the cell are going to go through the layers of the rough ER so that when it gets to the lipid membrane of the cell, it's going to be able to open up and leave. So this is the rough ER. So now we have the Golgi apparatus. We have left the ER and we're moving towards the UPS analogy. So when a protein is made in the ER, it's going to leave, and it's going to have a direction. It's going to go through this Golgi apparatus. It's going to go in the cis face, and it's going to leave through the trans face. So over here, these are your cis faces, and on the other side, the side that's facing out is the trans. I like to remember this as when proteins are leaving the Golgi apparatus, they're transitioning onto the next part of their life or their world. And so the transition or the trans phase is the leaving. And so as it's going through the Golgi apparatus is going to add phosphates, or it's going to add carbs like glycoproteins. This is going to modify where the protein goes or how it's used. Polysaccharides are going to be added on, but remember, not cellulose. That's the stuff that we find, that's the polysaccharide that we find in plants. And so the Golgi apparatus is composed of many hundreds of flattened cisternae. Cisternae are these flattened pancakes here. So we have 1, 2, 3, 4. These are going to be these flattened pancakes that allow the proteins to travel through from the cis to the trans. Lysosomes are our next adventure here, and we're starting to talk about these little bubbles that we have moving around our cell. And so lysosomes are filled with acids or hydrolytic enzymes that are going to break down, or remember, hydrolysis reactions are those that break down molecules. And so these lysosomes are going to hydrolyze any foreign bodies, damaged organelles, anything that is that needs to be broken down and reused. It's going to take it in through phagocytosis, digest it, and then those materials are going to be used again to serve as functions in the cell. And so this process is called autophagy. Remember that auto is a prefix for self, that the cell is breaking down the pieces and using them again to provide for itself. And a really interesting example is that a human liver cell will recycle half of its molecules each week. And so there is a lot of autophagy going on within our liver cells. And here we are at the very end talking about vacuoles. There are 3 different vacuoles that we're going to talk about. These are just these vesicles, these bubbles, these lipid membrane bubbles. So we have food vacuoles, which are going to form through phagocytosis and transport food to the lysosome, or transport stuff that needs to be broken down by the lysosome. We have contractile vacuoles, which are going to pump fresh water into or out of a cell, and it's going to help it to maintain a specific ion concentration. So if an organism is living in salt water and it needs to have a certain amount of fresh water within it, it's going to have those contractile vacuoles, which will pump water in or out. And finally, central vacuoles. We have large central vacuoles in plant cells, and they're going to hold that stuff that we call sap. And so as the amount of sap or water within the plant expands, you're going to have a larger central vacuole. And as the sap decreases, you're going to have smaller central vacuoles. And that's it. That is our endomembrane system, and I hope that helps.